I was about 10 years old when I first heard about Alzheimer's disease, when my grandfather, who was living in the United States, had developed the disease that caused him to lose his memories. Because of his deteriorating condition, my grandmother decided to send him back to the Philippines so we could look after him. 25 years later, I never thought that I'd be a scientist studying the disease that took my grandfather's life. Aging is the greatest risk factor, and it is estimated that 1 out of 9 people aged 65 and older has Alzheimer's disease dementia. In addition, the number of deaths from Alzheimer's disease continue to increase. With memory loss as the main symptom, Alzheimer's disease is a debilitating disorder that not only affects the patients, but can be very devastating for their caregivers and family members. My grandmother had to make the difficult decision to send my grandfather home back to the Philippines, where the cost of care was more affordable, and so that she could continue working to help the rest of us have a better life. My goal is to identify early changes in the structure of the brain to help develop therapeutics for preventing or slowing down the progression of Alzheimer's disease. Hi, my name is Kristeen, and I'm a second-year postdoc in Dr. Tara Tracy's lab at the Buck Institute. In this tiny lab talk, I will give an overview on Alzheimer's disease and what I'm currently working on to investigate the early changes that occur in the brain before Alzheimer's disease develops. So let's get started. Alzheimer's disease is the most common form of dementia, and it was named after a German psychiatrist, Dr. Alois Alzheimer. In 1901, Dr. Alzheimer had a patient who suffered from memory loss, sleep disorder, confusion, and aggressiveness. These conditions are now known to be symptoms of Alzheimer's disease. When she passed away 5 years later, Dr. Alzheimer looked at her brain under a microscope and he found accumulated proteins that have formed clumps or aggregates. These aggregated proteins are now known as plaques and tangles. Alzheimer's disease is a progressive neurodegenerative disease, which means that the disease destroys brain cells and it worsens over time. There is currently no cure for Alzheimer's disease, but scientists all over the world are trying very hard to study and understand the disease to find the cure. Although the cause of Alzheimer's disease is not completely understood, many scientists have looked at the aggregated proteins that Dr. Alzheimer discovered. Now we know that these proteins or aggregates are key players in disease progression. In our lab, we focus on studying one of the aggregated proteins found in Alzheimer's disease brains, called tau. Tau is a protein that is known to stabilize microstructures inside neurons. However, many factors such as stress, mutations, and modifications can cause tau to misbehave and stick together, forming clumps or aggregates. There's accumulating evidence now that the small aggregates of tau, also known as tau oligomers, are the toxic species of tau that can spread throughout the brain and cause neuronal death. We and other groups of scientists have shown that tau oligomers can damage the neuronal structures called synapses. Synapses are connections where signals are transmitted or passed from one neuron to another. These connections are key to brain's functions, such as learning and memory formation. Synapse loss is strongly linked to cognitive decline and memory loss in Alzheimer's disease. I hypothesize that synapse dysfunction caused by tau oligomers will eventually lead to neurodegeneration, and that maintaining synapse integrity is important and can prevent neurodegeneration. In our lab, we currently use many different tools to study the effect of tau oligomers on synapse function in human iPSC-derived neurons. These are neurons that we generate from adult human stem cells. Using modern biochemical techniques and cutting-edge mass spectrometry, we can study the molecular changes that occur in synapses in the presence of tau oligomers. We can also visualize the proteins in synapses and observe the changes in their expression using immunocytochemistry and confocal microscopy. In addition, we can measure how tau oligomers affect synaptic function using electrophysiology, which can measure electrical signals in synapses. With this research, we can identify early changes that occur in the synapses in the presence of tau oligomers even before synapse loss happens. These studies are necessary stepping stones for developing therapeutics for intervention or slowing down the progression of neurodegeneration in Alzheimer's disease.